Hello and welcome to part two, that is section B of the digestive system for Biology 112, AMP2. This time around, we are looking at the small intestines mostly in this section, and we're going to talk about some accessory organs as well. And then lastly, we'll do a run through the digestive process thus far. Remember to keep an eye on the big idea, and that is that these organs are highly and specially modified for digestion and absorption. And we're going to look into some of those finer details. Again, just to get our bearings straight, so far we've discussed the oral cavity, the accessory glands such as saliva and salivary glands that release the saliva, and we moved down through the esophagus. We discussed the stomach and how it begins protein and lipid digestion, and now we move into the small intestines. Later on, we finish up with the large intestines. So, where we left off was the stomach was releasing its contents through the pyloric sphincter, and here we enter the small intestines. This can be divided up into three main regions: the duodenum, jejunum, and the ileum, going from early, middle, to later sections respectively. Notice that the small intestine is going to finish when we enter the ileum, the ileocecal sphincter, and that ileocecal sphincter is going to be at the transition from the small intestine to the large intestines. Here in this image we see a cross section of the small intestines, and in the upper right perhaps you might observe these folds in the mucosa. We have a name for these, plicae circularis, in other words circular folds. These are permanent, half-inch tall folds, and these contain part of the submucosal layer. These are not found in the lower ileum, and they do not stretch out like the rugae in the stomach do. The main purpose for these structures are to increase surface area. Based upon these circular folds are smaller structures called villi. These villi would be barely, if not at all, visible in the above picture, so we need to refer to the diagram below to show the structure of the villi. These are approximately 1 millimeter tall, and keeping in mind that these are part of the mucosal layer, and therefore this mucosal layer has a layer of lamina propria as a basement membrane for the epithelia to attach to. If we look more closely in the villi, we see that there are capillaries running through here. These are both vascular capillaries in terms of blood, and then also the green ones indicate lacteals, the lymphatic capillaries. A lacteal is a sort of lymphatic capillary within the villus, and it is responsible for absorbing liquids and some lipid soluble vitamins. And then if we look closely on the tip of this villus is one individual cell. So we see it's an individual cell because of the presence of the purple nucleus. On the border of this cell are microvilli. And again, this is a very microscopic level that we're working with. So this microvilli is also called brush border, and that is because the quote-unquote hair-like structures that exist on the edge of this. Embedded deep within these fine cilia-shaped objects that come out of the cell are enzymes embedded in there, and those enzymes are going to help break down digestion further. So in summary, the surface area of the small intestines is quite great. When we A, consider the length of the small intestines, B, consider the circular folds, C, consider the villi, and D, consider the microvilli, perhaps now we gain an appreciation how the small intestines has a fantastic amount of surface area. Here is another image showing the structures that we just discussed. It is noteworthy that this picture also shows some pits in the epithelia. So in addition to the villi, we have pits that are lined with intestinal crypts and enteroendocrine cells, T cells, and Paneth cells. We'll get into those more so later on. In the submucosa, we see areolar connective tissue, and later on into the small intestine, we're going to see Peyer's patches exist in that submucosa. These have lymphatic properties. And lastly, we'll see duodenal glands in the duodenum region only. Now we take a closer look at some of these specialized intestinal glands. So let's start by getting our bearings straight. Remember that we are in the small intestine, and these finger-like projections are referred to as villi. And so notice that we're discussing these cells that line the sides of these villi. The first one to note is the mucosa layer and the submucosa layer that has the vessels running through it. And these are going to secrete some intestinal juice. And we see that goblet cells have the function of producing mucus. This mucus is going to lubricate and protect the intestinal lining. And in addition to the goblet cells, we're going to have absorptive cells. These are going to digest and absorb the nutrients that we've been discussing. And we also have enteroendocrine cells. And these enteroendocrine cells are also called G cells. In the stomach, we see that this secretes gastrin into the blood. Well, in these intestines, we see that these enteroendocrine cells secrete a substance called secretin. This is a hormone. And then also cholecystokinin is, by the way, abbreviated by CCK. These together are going to be hormones that decrease stomach motility and also secretion activities. And the next cell we might see in these intestinal glands are going to be Paneth cells. These are going to secrete lysozyme to kill bacteria. Lysozyme is a sort of enzyme that is able to break down the foreign bacteria. And now we take a break from our discussion about the small intestines. And by the way, keep in mind the big idea behind the small intestines is absorption of nutrients. And as we switch gears, we're now talking about some accessory organs. We're discussing the liver and gallbladder and how these help the digestive system do its work. We see that the liver is quite a large large organ in the human body. And upon some gross anatomy, we can observe that it has a left and a right lobe, and these are separated by a falciform ligament. And buried deep into this liver is a structure greenish in color. That's going to be the gallbladder. That's a sac that stores bile. And the liver has multiple functions. The liver has many functions such as carbohydrate, lipid, and protein metabolism. In other words, we're talking about breaking down these substances for digestion. The liver also removes drugs and hormones from the blood. It excretes bilirubin, synthesizes bile salts, stores vitamins and minerals. It also has the task of bacterial phagocytosis and also the activation of vitamin D. So it's a wide-ranging organ in its functions, but here we focus mostly on the synthesis of bile salts. So in summary on this slide, we see we're going to be talking about three main organs to help us out here soon. The liver, the gallbladder, and later on we see the pancreas are all going to secrete fluids into the duodenum of the small intestines. Let's look at some histology of the liver. Notice that we're going in depth into one section of that liver. Upon one magnification, we see these hexagonal shapes, and that central dot is called our central vein. And in image B, we zoom in a little bit more and see the hexagonal shape a little bit closer. Now let's move into the diagram below. In this diagram, we see that hepatocytes are arranged in these lobules. So this one hexagonal shape is going to be called a lobule. So these are going to also be called, these hepatocytes are also going to be called liver cells. And in addition, in this diagram, we see portal triads. Triads with the prefix meaning 3. So we're looking for 3 structures. That's going to be composed of a hepatic duct, a bile duct, and branches of the hepatic portal vein and hepatic portal artery. They're going to be located on the periphery of each lobule. So at the center of each one of these lobules, we see that central vein that's going to merge to form numerous hepatic veins. And in addition, we see hepatic sinusoids. These are very permeable capillaries. This is where nutrients are absorbed into the hepatocytes. Along these sinusoids, we're also going to see macrophages. Kupffer cells are going to help us kill certain bacteria. Upon closer inspection, this slide shows bile production and bile function. And this bile is secreted by hepatocytes. And notice that the bile passes into the bile canaliculi to bile ducts, and this is all coming from the sinusoids within the lobule of the liver. Bile is an alkaline fluid, and its function is the mechanical digestion of lipids. Here is a histology image showing the gallbladder. Notice we see some simple columnar epithelium, 3 layers of smooth muscle, and a serosa. It functions in the terms of the gallbladder is in charge of bile storage. That is not bile production, bile storage. If the bile is not immediately needed for digestion, it stores it and concentrates it. Sometimes the cystic duct that comes from the gallbladder can be blocked. In other words, these salts will sometimes crystallize and become too concentrated. We therefore will get a gallstone, and that can cause jaundice, as shown in the lower right. In this slide, we take a look at how the blood, or I should say the liver, is supplied with blood. And let's do a review of some of the things we've done in lab. In lab class, we had a dissection where we tracked down the hepatic portal vein, and that hepatic portal vein is going to receive a nutrient-rich blood from the stomach, spleen, and intestines. And then we have the hepatic artery, which is going to branch off from the aorta. So that means the hepatic artery carries oxygenated blood to the liver, and the hepatic portal vein carries nutrient-rich but oxygen-poor blood from the capillary beds of the GI tract, the spleen, and the pancreas to the liver. Notice then we bring this material to the liver sinusoids, which is then going to go to the central vein, hepatic vein, and into the main inferior vena cava, and then to the right atrium of the heart for a circuit around the circulatory system. So in summary, the liver both gets blood to make it run, and then also it gets blood to filter. Let's look at another accessory organ to digestion, and now we look at the pancreas. That means we're looking at this yellow object here. We see that in some gross anatomy, this pancreas has a region called the head, and then it has a body, and then a tail. In terms of general function, the pancreas has both endocrine and exocrine functions. So when we say endocrine, we're talking about the endocrine system that is releasing hormones. So endocrine cells of the pancreas produce and secrete the hormones insulin and glucagon, whereas the exocrine cells of the pancreas produce pancreatic juice. And by the way, in a minute, we're going to go into the acinar cells that produce some enzymes as well. This slide serves to just be a nice overview of the ducts. And so that means we're talking about ducts that are emptying into the duodenum. And we see that the right and left hepatic duct form to make a common hepatic duct. And then we are getting a cystic duct from the gallbladder to a common bile duct. And keep in mind, we're all talking about secretions, fluids being injected into the duodenum. And then in addition to this bile that we're receiving from either liver or gallbladder, then we're also injecting pancreatic juice from the pancreas. Here we see some histology images and a matching diagram. We see these acinar cells, and these are going to produce hydrolytic enzymes. These consist of 99% of this gland. However, we have little quote-unquote islands within these acinar cells. Notice that these are called islets of Langerhans. Islets meaning tiny islands. These are only about 1% of the pancreas gland. These are going to be pale stained, as we can see below. And these are going to produce hormones that we discussed earlier. And now we have some information on that pancreatic juice. Interestingly, we produce about 1.5 quarts a day. And this pancreatic juice is going to be having a pH of 7.1 to 8.2. Remember that a pH of 7 is neutral. And we're looking at water, enzymes, and sodium bicarbonate being produced. And in addition, we're going to have some broad range of digestive enzymes. And we can see that, again, these digestive enzymes all serve to break down the foodstuffs. Keep in mind that this foodstuff at this point is not called food. We call it chyme because it has received chemicals from the stomach and also received mechanical digestion from the stomach as well. So that does it for the anatomy and function of the small intestines. And in summary, I hope we saw that the specialized organs are unique in their design to A, help us with secretion, and B, help us with the absorption of nutrients. So now we switch gears a little bit, and we're going to talk about the digestive process. And we're going to pick up from where we left off the last time. The last time we took a journey from the oral cavity to the stomach. And so now we're going to view the digestive process in terms from the stomach to the small intestines. So think about eating a meal. That meal travels through peristalsis down into the stomach and spends about 4 hours in there. That rate of emptying from the stomach depends on the contents of the duodenum. And the chyme, as we call it now, stretch, and these chemoreceptors will target reflexes that inhibit gastric secretions and prevent further duodenal filling. In terms of the substances in the former food, carbohydrates, as opposed to proteins and fats, move through the quickest. Fats are digested more slowly. These may linger in the stomach for 6 or more hours. And so after the food, quote-unquote food, moves to the small intestines, this is where most of the absorption takes place. And keep in mind we have some mechanical processes occurring here and some chemical processes occurring here. Mechanical digestion and chemical digestion is shown in this overview. Showing as an image in that mechanical digestion, we see this chyme being worked through the alimentary canal. One process that works it through in mechanical digestion is segmentation. That is a major movement of the small intestine, and that's going to be a localized contraction in areas containing food. Then we also have peristalsis, and that is a sort of wave that propels the chyme onward through the intestinal tract. So our image below is going to show segmentation. The image up top is going to show peristalsis. Weak peristalsis happens in the small intestine compared to the stomach. The chyme remains in the small intestines for about 3 to 5 hours. In this time, we also have more segmentation occurring, and we're going to get the local mixing of chyme with some of those intestinal juices. This diagram serves to go a little bit more in depth into the chemical digestion occurring in the intestine. And so you might recall that we had a wide variety of secretions that enter the duodenum. And so these secretions, this intestinal juice, provides a vehicle for absorption of substances from that chyme as they come in contact with the villi. So we see on this image on the right, these projections are trying to show the villi. And so in the blue, we would have intestinal lumen, and in the brown, we're going to have tissue, and you can see at the bottom is the capillaries. Keep in mind that what we're viewing in this diagram is the ultimate goal of the digestive system. In other words, these organs in the human body in general need these essential building blocks. And here we see these essential building blocks entering the bloodstream. Building blocks such as amino acids coming in, or glucose coming in. These are all the essential things that the human body needs. Well, some intestinal enzymes, for example, the brush border enzymes, are going to break down these carbs and proteins on the surfaces of that epithelia of microvilli. We're going to get a little bit of digestion occurring in the lumen of the small intestine as well. So these larger carbohydrates get broken down into smaller monosaccharides for absorption. And these larger proteins get broken down into polypeptide chains and then ultimately tiny little amino acids. So let's take, for example, lactase. Lactase is a larger carbohydrate that breaks down, let me back up. Lactase is an enzyme that breaks down lactose sugar. And so that lactose sugar needs to be broken down into smaller subunits like glucose. As we can see in the lower right, a person who might be lactose intolerant is unable to break down that larger lactose into the tiny subunits. Let's now take a look at chemical digestion further in terms of bile and what that purpose is. We see that bile is a yellow-green sort of alkaline substance. The color comes from pigments that are in there. We also have salts in this bile, cholesterol in this bile, triglycerides, phospholipids, and electrolytes exist in this bile. The purpose of the salts is to emulsify fats, in other words, break them down and help us with digestion. And here we take a look at the small intestine chemical digestion some more. We're looking at enzymes and bicarbonate. In review, a couple slides ago, we saw that the pancreas releases a wide, broad spectrum of digestive enzymes, shown in the upper right. And then also we're going to get bicarbonate secreted as well. This bicarbonate tends to be a neutralizer. So think about Tums, an antacid tablet, having a lot of bicarbonate in there. It's going to take a lot of acid in the stomach, and by adding bicarbonate, we neutralize that acid. This diagram shows step number 6 of the digestive process overall. So after this food is digested into those tiny monomers, absorption can take place. Again, most of this absorption is occurring in the duodenum, but it can't occur anywhere else in the small intestines. We're going to get a little bit of reclamation of the bile salts in the ileum. So notice that we have pointed out that we do not have to know details of all these, but essentially the whole overview is that once those tiny building blocks are broken down, as shown on the left side, we see that we can get them into a blood capillary of a villus. And this can occur multiple ways. Number one, the most easy is through diffusion, and that's an energy-free process. But then we have number two, facilitated diffusion, where we require a little bit of energy to move that molecule through. Osmosis is another process which can move water through. And then lastly, active transport is not as common, but still requires energy to move some of these larger, more complex molecules into that membrane. Our last step in the process is that what is left over is delivered to the large intestine. And that's going to happen through the ileocecal valve. The next section, we get more in depth into that large intestine. This diagram simply serves to show that pathway of the nutrients getting into the bloodstream. Again, this finger-like projection being the villus, showing capillaries receiving those tiny building blocks. And so we now switch gears, and after talking about that steps of digestion, we now have some extra information, a sort of appendix to this discussion. By the way, that's not the appendix organ. We're talking about a textbook organization here. So over the next few slides, we talk about digesting carbohydrates, proteins, and lipids. So perhaps if you go to your cabinet, if you're studying at home right now, you can pull out any food stuff, a can of food, a box of food, and we can look at the nutrition facts and see that food is mostly carbohydrates, lipids, and proteins. So essentially, we're throwing a whole bunch of carbohydrates, proteins, and lipids into this body. Let's go see how each one of those gets broken down. So in this slide, we're talking about carbohydrates. And in terms of food, you're probably aware of carbohydrates being things like pastas, fruits, grains, etc. This is how we break those down. First step is in the mouth. These carbohydrates are greeted with salivary amylase. Then you might remember the food moves through the esophagus and the stomach. I should say bolus, I suppose. Nothing happens at this point to the carbohydrates. Then in the duodenum, we are receiving juices from the pancreas. Pancreatic amylase is an enzyme that's going to help break down carbohydrates. And then as we move further along the small intestines, we encounter brush border enzymes, and these are going to act upon the disaccharides. Disaccharide being a slightly larger molecule, we then break that down into smaller monosaccharides, such as fructose, glucose, and galactose. So again, if you looked on the nutrition facts of a can of soda, you'll see the number 2 ingredient is typically high fructose corn syrup. We're talking about very simple monosaccharides. As we mentioned earlier, some persons are lactose intolerant. That means genetically they're not able to produce an enzyme, and that the enzyme is going to break down those sugars. So the problem is this is going to create gas and diarrhea. So in summary, those are some of the steps to break down carbohydrates. Now we look at how we're going to break down those proteins in food. So by this, we're talking about things like meats and beans and nuts, etc. So in the oral cavity and esophagus, nothing happening. And then we arrive at the stomach, where you might recall that hydrochloric acid is going to denature and unfold those proteins. We have then pepsin turning proteins into polypeptides. Furthermore, proteins get broken down in the pancreas because that is secreting, I should say, it's secreting digestive enzymes, and these enzymes are going to split the peptide bonds that hold those amino acids together. Another way we get proteins broken down is through brush border enzymes, and these enzymes are going to again break those peptide bonds, but these are the ones that attach the terminal amino acids to the carboxyl ends of the peptides. And then we see that in the large intestines, nothing happening with the digestion of proteins. In terms of fats, in other words, lipids, we think about, well, cheeses perhaps, or oils or butters having a large amount of lipids. And so the digestion of these begin in the mouth. Here we have lingual lipase. And again, in the esophagus, not much happening because most lipid digestion is occurring in the small intestines. We're going to get emulsification of that fat thanks to bile and globules of triglycerides. Also, these lipids are going to be broken down thanks to pancreatic lipase. That's going to split these triglycerides into fatty acids and monoglycerides. In the brush border cells, however, we do not have enzymes that break down lipids. As a sort of addition, we also have the ability to break down nucleic acids. Nucleic acids are handy for the body as well, even though we do not see them on a foodstuff, a nutrition facts label. So these nucleic acids get broken down into individual nucleotides for absorption. So when we say nucleotides and nucleic acids, perhaps you know that we're talking about things like DNA and RNA. So the pancreatic juice is going to have two enzymes that break down nucleic acids. One is ribonuclease, which digests ribonucleic acid, and deoxyribonuclease, which digests deoxyribonucleic acid, in other words, DNA. These nucleotides produced are furthermore digested by brush border enzymes, and these are going to be absorbed into the human body thanks to active transport. So in this table here, we see a nice summary of those enzymes that are secreted and their actions that they do. And then lastly, we start wrapping up our discussion by talking about digestive hormones. What are some important hormones that have functions for digestion? We start with gastrin. Gastrin is going to be in the stomach, and we find that in the gastric and ileocecal sphincters. Gastrin is a hormone that stimulates stomach secretions and motility. Another hormone secreted is going to be gastric inhibitory peptide, abbreviated by GIP. That's going to be found in the stomach and the pancreas. And here we're going to be inhibiting the activities of the stomach. Another hormone found in the digestive system is going to be secretin. We're going to find this in the pancreas, liver, and stomach. And secretin is going to be released from the small intestines in response to an increase in chyme acidity. So the secretin causes the release of an alkaline solution from the liver and pancreas, and that helps neutralize the acidic chyme. And the fourth hormone we discussed is cholecystokinin, abbreviated by CCK. We'll find this in the pancreas, gallbladder, sphincter of Oddi, and stomach. CCK is a hormone released from the small intestine, and that's in response to fatty chyme. So the CCK is going to stimulate smooth muscle contractions in the gallbladder, releasing bile into the duodenum. It also stimulates the pancreas to release pancreatic juice, and relaxes the hepatopancreatic ampulla. So that does it for the small intestines and some digestive processes. This is concluding section B of our discussion. Next video is going to be discussing section C of the digestive system, where we go into more in depth into the large system, the large intestine, and see how it helps us with the digestive system.